Next topic, second messengers. Second messengers are intracellular signaling molecules that are produced in response to the binding of a ligand to a membrane-bound receptor. They mediate intracellular signaling cascades that regulate a wide range of cellular functions such as gene expression, metabolism, proliferation, differentiation, and apoptosis. Second messengers include cyclic adenosine monophosphate CAMP, inositol triphosphate IP3, diacylglycerol DAG, calcium ion Ca2+, and nitric oxide NO. CAMP is synthesized by adenylate cyclase and is degraded by phosphodiesterase. It regulates various cellular processes including glucogenolysis, gluconeogenesis, and lipolysis. Examples of receptors that activate CAMP include beta-adrenergic receptors, glucagon receptors, and parathyroid hormone receptors. Next, IP3. IP3 is produced by the cleavage of phosphatidyl inositol 4,5 biphosphate PIP3 by phospholipase C PLC. It binds to IP3 receptors on the endoplasmic reticulum and triggers the release of Ca2+. It regulates various cellular processes including muscle contraction, exocytosis, and cell division. Examples of receptors that activate IP3 include GQ-coupled receptors such as alpha-adrenergic receptors and muscarinic acetylcholine receptors. Then DAG. DAG is produced by the cleavage of PIP3 by PLC. It activates protein kinase C, PKC, which regulates various cellular processes including gene expression, cell growth, and apoptosis. Examples of receptors that activate DAG include GQ-coupled receptors such as alpha-adrenergic receptors and muscarinic acetylcholine receptors. Ca2+ is one of the most widely used second messengers. It is released from intracellular stores such as ER or the mitochondria or enters the cell through plasma membrane channels. It regulates various cellular processes including muscle contraction, exocytosis, gene expression, and apoptosis. Examples of receptors that activate Ca2+ include GQ-coupled receptors such as alpha-adrenergic receptors and muscarinic acetylcholine receptors. Then nitric oxide. Nitric oxide is produced by nitric oxide synthetase and acts as a vasodilator and a neurotransmitter. It regulates various cellular processes including blood pressure, immune response, and neurotransmission. It activates guanylate cyclase and increases the levels of cyclic guanosine monophosphate, which in turn regulates various cellular processes. Examples of receptors that activate NO include the NMDA receptor and the muscarinic acetylcholine receptor. Overall, second messengers play a crucial role in intracellular signaling cascades and regulation of cellular functions. Dysregulation of second messenger signaling has been implicated in a wide range of diseases including cancer, cardiovascular disease, and neurological disorders. Then proteoglycans, glycosaminoglycans. In detail, proteoglycans and glycosaminoglycans or GAGs are types of large complex biomolecules that are important components of the extracellular matrix, a network of macromolecules that supports and regulates cell behavior. Then proteoglycans are composed of a core protein and one or more long linear chains of GAGs, which are highly charged sulfated polysaccharides. GAGs are repeating disaccharide units composed of an amino sugar, either N-acetyl glucosamine or N-acetyl galactosamine, and a uronic acid, either glucuronic acid or iduronic acid. The GAG chains of proteoglycans are attached to the core protein through a covalent linkage to the specific serine residue. GAGs can also exist independently of proteoglycans and they play important roles in various biological processes. They can interact with a variety of proteins such as growth factors, cytokines, and enzymes and regulate their activity, localization, and stability. The unique properties of GAGs, such as their highly negative charge and hydrophilicity, make them ideal for binding and sequestrating cations and water, which can help maintain tissue hydration and osmotic balance. Some major examples of proteoglycans and GAGs include hyaluronic acid, a large non-sulfated GAG that is a major component of ECM and synovial fluid and is involved in lubrication, shock absorption, tissue repair. Then chondroitin sulfate, a sulfated GAG that is found in cartilages, bone, skin, and blood vessels and is important for structural integrity and resilience. Then heparin sulfate, a highly sulfated GAG that is present on the surface of most cells and is involved in cell signaling, adhesion, and migration. Then dermatan sulfate, a sulfated GAG that is abundant in skin, tendons, and blood vessels and is important for elasticity and mechanical strength. Overall, proteoglycans and GAGs are critical components of the ECM and play important roles in tissue development, homeostasis, and repair. They also have a variety of functions outside the ECM, such as regulating the activity of signaling molecules and protecting tissues from damage. Then, blood grouping in detail. Blood grouping is the classification of blood based on the presence or absence of certain antigens and antibodies on the surface of red blood cells. There are 4 main blood groups: AB, and O. Each of these groups is determined by the presence or absence of specific antigens on the surface of the RBCs. The presence of blood grouping involves 2 main steps: blood typing and cross-matching. Blood typing. Blood typing involves determining the ABO and RH blood groups of an individual. This is done by using specific antibodies that react with the antigens on the surface of the RBCs. Then ABO blood group system. The ABO blood group system is based on the presence or absence of 2 antigens, A and B. Individuals with blood group A have antigen A on their RBCs, while those with blood group B have antigen B on their RBCs. Those with blood group AB have both A and B antigens on their RBCs, while individuals with blood group O do not have either A or B antigens on their RBCs. RH blood group system. The RH blood group system is based on the presence or absence of the RH antigen on the surface of the RBC. Individuals who have the RH antigen are considered RH positive, while those who do not have the antigens are RH negative. Then cross-matching. Cross-matching is the process of mixing a sample of the recipient's blood with a sample of the donor's blood to determine if they are compatible. This is done by checking for agglutination for complete clumping of the RBCs. If agglutination occurs, it indicates that there is a mismatch and the blood is not compatible. Importance of blood grouping. Blood grouping is important for several reasons, including blood transfusions. Blood grouping is essential for ensuring that the recipient receives compatible blood during a blood transfusion. Then pregnancy. Blood grouping is important during pregnancy to determine the risk of hemolytic disease of the newborn, a condition where the mother's antibodies attack the RBCs of the fetus. Then forensic investigations. Blood grouping can be used in forensic investigations to help identify individuals based on the blood found at a crime scene. Overall, blood grouping is a crucial aspect of modern medicine as it helps ensure the safety and well-being of patients who require blood transfusions and can also provide important information in the field of forensic science. Then order of permeability. The order of permeability is a classification of substances based on their ability to pass through biological membranes. The order of permeability can vary depending on the type of membrane, but in general it follows the pattern of lipids, small uncharged molecules, small polar molecules, large uncharged molecules, and ions. Lipids. Biological membranes are composed primarily of lipids and lipid-soluble substances such as gases O2, CO2, and N2, and hydrophobic molecules like steroids, fatty acids are highly permeable through these membranes. Then small uncharged molecules. Small uncharged molecules such as water, ethanol, and urea can pass through membranes through simple diffusion. Small polar molecules. Small polar molecules such as glycerol and ethanolamine can also pass through membranes but at a slower rate compared to uncharged molecules due to the polar head of the lipid bilayer. Then large uncharged molecules. Larger uncharged molecules such as glucose and sucrose are less permeable through membranes compared to smaller uncharged molecules. So first, lipids. biological membranes are composed primarily of lipids, and lipid-soluble substances such as gases O2, CO2, N2, and hydrophobic molecules like steroids, fatty acids are highly permeable through these membranes. Then small uncharged molecules are there. So the small uncharged molecules such as water, ethanol, and urea can pass through membranes through simple diffusion. Then small polar molecules such as glycerol and ethanolamine can also pass through membranes but at a slower rate compared to unchanged molecules due to the polar head of the lipid bilayer. Then large uncharged molecules. Larger uncharged molecules such as glucose and sucrose are less permeable through membranes compared to smaller uncharged molecules and require the assistance of transport proteins. Then ions. Charged molecules such as ions are generally not permeable through biological membranes as their charges interact with the charged polar head of the lipid bilayer. However, ions can pass through ion channels, which are specialized protein structures in the membrane that facilitate the movement of specific ions. The importance of understanding the order of permeability lies in its implications for drug delivery and design. For example, drugs that are lipid soluble are more likely to cross biological membranes and reach their targets, while drugs that are polar or charged may require a transport protein or other delivery mechanism. The order of permeability can also be used to predict the movement of molecules to different membranes in the body and to understand the mechanisms of absorption and secretion. Then order of permeability, once again, refers to the relative permeability of different substances across the biological membrane. It determines which molecule can pass through the membrane and at which rate, which is essential for the proper functioning of cells and tissues. Then this order of permeability of substances across biological membrane also follows the following sequence, like small nonpolar molecules. Small nonpolar molecules such as oxygen and carbon dioxide are highly permeable across biological membranes due to their small size and nonpolar nature. They can easily diffuse to the lipid bilayer of the membrane. Then small uncharged polar molecules. Small uncharged polar molecules such as water and ethanol can also diffuse across biological membranes, but their permeability is lower than that of small nonpolar molecules. This is because they are polar and cannot easily pass through the nonpolar lipid bilayer. However, the small size allows them to pass through specialized channels in the membrane. Then large uncharged polar molecules. Large uncharged polar molecules such as glucose and sucrose have a lower permeability than small uncharged polar molecules. They cannot diffuse through the lipid bilayer and must rely on specialized transporters or channels to cross the membrane. Then ions such as sodium, potassium, and chloride are highly charged and cannot pass through the nonpolar lipid bilayer. They must rely on specialized channels or transporters to cross the membrane. The order of permeability is important for understanding how molecules are transported across biological membranes and how this can be regulated. It is also important in drug development as the permeability of a drug molecule across biological membranes affects its ability to reach its target and exerts its effects. For example, lipid-soluble drugs are generally more permeable across biological membranes and can reach their targets more easily than water-soluble drugs. This knowledge can be used to design more effective drugs with better permeability and bioavailability. Then lectins. Lectins are a diverse group of carbohydrate-binding proteins found in plants, animals, and microorganisms. They have the ability to recognize and bind to specific carbohydrate molecules structures such as glycoproteins, glycolipids, and polysaccharides. The binding is typically reversible and can occur with high specificity and affinity. Lectins play important roles in various biological processes such as cell adhesion, signaling, and immune response. They can also have therapeutic applications such as in cancer treatment and diagnosis. Then here are some examples of lectins. Concavalin A. This is a lectin found in jack bean like Canavalia ensiformis. It is widely used in biochemistry and cell biology for its ability to bind to specific carbohydrates and facilitate their isolation and purification. Then wheat germ agglutinin. This is a lectin found in wheat germ, Triticum aestivum. It has been shown to have anti-tumor and antiviral properties and is used in research to study cell adhesion and signaling. Then galectins. These are a family of lectins found in many different tissues and organs. They play important roles in cell adhesion, signaling, and immune weapons. Immune response, and have been implicated in various diseases such as cancer and autoimmune disorders. Then resin is a highly toxic lectin found in the seeds of castor oil plant, Ricinus communis. It is considered a bioterrorism agent due to its ability to inhibit protein synthesis and cause cell death. Overall, lectins are important biomolecules with diverse functions in various biological processes. Their ability to recognize and bind to specific carbohydrate structures makes them valuable tools in research and potentially useful in therapeutic applications. Then, inborn errors of metabolism. Inborn errors of metabolism refer to genetic disorders that affect the metabolism of specific biomolecules such as carbohydrates, proteins, and fats. These disorders are usually caused by a deficit or defect in an enzyme or cofactor required for a normal metabolism of these biomolecules. There are many different types of inborn errors of metabolism, each with a specific symptom, causes, and treatment. Some of the most common types are phenylketonuria, PKU. This is a genetic disorder that affects the metabolism of the amino acid phenylalanine. People with PKU are unable to properly metabolize phenylalanine, which can cause a buildup of toxic byproducts in the body. Symptoms of PKU can include developmental delay, intellectual disability. Symptoms of PKU can include developmental delay, intellectual disability, and seizures. The condition can be treated by avoiding foods that contain phenylalanine and by taking supplements of other amino acids. Then galactosemia. This is a genetic disorder that affects the metabolism of sugar galactose. People with galactosemia are unable to properly metabolize galactose, which can cause a buildup of toxic byproducts in the body. Symptoms of galactosemia can include jaundice, poor feeding, and intellectual disability. The condition can be treated by avoiding foods that contain galactose. Then maple syrup urine disease. This is a genetic disorder that affects the metabolism of branched chain amino acids. People with maple syrup urine disease are unable to properly metabolize these amino acids, which can cause a buildup of toxic byproducts in the body. Symptoms of this condition can include poor feeding, lethargy, and seizures. The condition can be treated by restricting the intake of these amino acids and by taking supplements of other amino acids. Tay Sachs disease. It is a genetic disorder that affects the metabolism of a type of fat called ganglioside. People with Tay Sachs disease are unable to properly metabolize this fat of ganglioside, which will definitely cause a buildup of toxic byproducts in the body. Symptoms of the condition can include developmental delay, intellectual disability, and seizures. This is currently no cure for Tay Sachs disease, and treatment is focused on managing symptoms. Then Gaucher's disease. This is a genetic disorder that affects the metabolism of a type of fat called glucocerebroside. People with Gaucher disease is unable to properly metabolize this fat, which can cause a buildup of toxic byproducts in the body. Symptoms of the condition can include bone pain, fatigue, and anemia. The condition can be treated with enzyme replacement therapy. Then inborn errors of metabolism are typically diagnosed through blood and urine tests that measure the levels of specific biomolecules in the body. Treatment often involves a combination of dietary changes, supplements, and medications to manage symptoms and prevent complications. It's important to note that the treatment options for inborn errors of metabolism vary depending on the specific condition and its severity. Early diagnosis and treatment are crucial for managing symptoms and preventing long-term complications. The Nobel Prize studies life science in detail. The Nobel Prize is one of the most prestigious awards given to individuals who have made exceptional contributions in various fields of science, literature, and peace. In the field of life science, there have been numerous Nobel Prize laureates who have made significant contributions to various areas of research. Here are some of the major Nobel Prize studies in life science along with their contributions. Then James Watson and Francis Crick in 1962. These two scientists discovered the double helix structure of DNA, which is the fundamental basis for the storage and transmission of genetic information. Then Frederick Sanger in 1958 to 1980. Sanger developed methods for determining the sequence of amino acids in proteins and the sequence of nucleotides in DNA. These techniques revolutionized the field of molecular biology. Then Linnaeus Pauling was awarded the Nobel Prize for his work on the nature of chemical bonding and structure of molecules. He made many important contributions to the understanding of protein structure, and was the first to propose that sickle cell anemia was a molecular disease caused by a genetic mutation. Then Barbara McClintock in 1983 was awarded the Nobel Prize for her discovery of transposable genetic elements or jumping genes which can move around within the genome and have important implications for genetic inheritance and evolution. Then Gregor Mendel, posthumously 1901. Mendel was a pioneering geneticist who studied the inheritance of traits in pea plants and formulated the basic laws of genetics. Paul Berg in 1980. Berg was awarded the Nobel Prize for his development of recombinant DNA technology, which allows scientists to manipulate and analyze genes in a way that was previously impossible. Then Joshua Lederberg in 1958. Lederberg was awarded the Nobel Prize. Wait a minute. Once again, we are going to learn about the Paul Berg in 1980. Berg was awarded the Nobel Prize for his development of recombinant DNA technology by Paul Berg, which allows scientists to manipulate and analyze genes in a way that was previously impossible. Then Joshua Lederberg was awarded the Nobel Prize for his work on bacterial genetics and the discovery of mechanisms of genetic recombination in bacteria. Then Sidney Brenner in 2002. Brenner made important contributions to the field of molecular biology, including the discovery of messenger RNA and the use of the nematode Xenorhabditis elegans and as a model organism for studying developmental biology. Hargobind Khorana was awarded the Nobel Prize for his work on the genetic code and the synthesis of nucleotides in 1968. Genetic code and synthesis of nucleotides. Rosalind Franklin, posthumously in 1962. Franklin made critical contributions to the discovery of double helix structure of DNA through her work on the X-ray crystallography, but was not recognized with the Nobel Prize due to her untimely death. These Nobel Prize studies have had a profound impact on the field of life science and continue to inspire and inform current research efforts. Next, biostatistics, t-test, ANOVA, STSE variants, real life use. Biostatistics is an important field in life science that involves application of statistical methods to analyze and interpret data from biological experiment. Some of the commonly used statistical texts in biostatistics include t-tests, ANOVA, analysis of variance, standard deviation, ST, standard error, and variance. Here are some real-life examples of their use and importance. T-test. The t-test is used to determine if there is a significant difference between the means of two groups of data. For example, in a clinical trial, a t-test can be used to compare the effectiveness of new drugs versus a placebo. Another example is comparing the mean weight of a certain group of animals before and after a treatment. Then ANOVA. ANOVA is used to compare the means of more than two groups of data. For example, ANOVA can be used to compare the effectiveness of 3 different treatments on a certain disease. Another example is comparing the mean growth rate of different plant species under different conditions. Then standard deviation or ST is a measure of the amount of variation or a dispersion of a set of data from the mean. It is used to assess the precision of the data. For example, if the ST of a dataset is large, it suggests that the data points are more widely spread out around the mean, indicating less precision. Then standard error. Standard error is a measure of the precision of the mean. It represents the variation of the sample means around the population mean. It is often used to estimate the confidence interval of the mean. For example, in a clinical trial, the SE can be used to estimate the 95% confidence interval for the mean difference between the treatment and control groups. Then variance. Variance is a measure of how much the data deviates from the mean. It is used to assess the spread or dispersion of the data. For example, in a study on the effect of temperature on plant growth, variance can be used to determine how much the growth rate varies with different temperatures. In summary, biostatics plays a crucial role in analysis and interpretation of data in life science. So, once again, we will be learning about the standard deviation, amount of variation or dispersion of a set of data from its mean. Then standard error is a measure of precision of the mean. Then variance is a measure of how much the data deviates from the mean. Major discoveries in biology, contributors name latest. There have been numerous major discoveries in biology over the years, and it's difficult to name them all. However, some of the most significant discoveries and their contributors include discovery of the structure of DNA, James Watson and Francis Crick; discovery of natural selection, Charles Darwin; discovery of the cell, Robert Hooke and Anton van Leeuwenhoek; discovery of the laws of heredity, Gregor John Mendel; then discovery of the theory of evolution, Charles Darwin; discovery of penicillin, Alexander Fleming; discovery of the structure of protein, Linnaeus Pauling; discovery of the genetic code, Marshall Nirenberg, Hargobind Khorana and Robert Hawley; discovery of restriction enzymes, Werner Arber, Daniel Nathans and Hamilton Smith; discovery of RNA interference, RNAi, Andrew Fire and Craig Mello. Here are just a few of the many significant discoveries made in biology. Then 2021 Nobel winners in physiology or medicine was awarded jointly to 3 scientists for their discoveries of the molecular mechanisms controlling the circadian rhythm. The laureates are Dr. Jeffrey C. Hall, who is an American citizen born in New York, USA, who is an emeritus professor at the University of Maine in Orono, Maine, USA. Dr. Michael Rosbash, American citizen, born in Kansas City, Missouri, USA, who is a professor at Brandeis University in Waltham, Massachusetts, USA. Dr. Michael W. Young, American citizen, born in Miami, Florida, USA, who is a professor at Rockefeller University of New York. Emeritus professor at the University of Maine, then University of Waltham, Rockefeller University of New York. These 3 scientists were awarded the Nobel Prize for their work on the molecular mechanisms that control the circadian rhythm or the body clock. Their discoveries have helped to explain how plants, animals, and humans adapt their biological rhythms to the Earth's rotation. Their research has also shed light on the mechanisms that underlie sleep disorders, jet lag, and other health issues related to the disruption of circadian rhythm. Hall, Rosbash, and Young used fruit flies as a model organism to identify the genes that control the circadian rhythm. They discovered a feedback loop involving proteins that regulate the activity of these genes, which allows the body to maintain a stable internal clock. Their research has also led to the development of new treatments for sleep disorders and their circadian related health problems. So, with the help of this video, we have completed the whole syllabus of NET paper 2, just by having a quick recap about all this topic. So we have already completed the paper 1, quick summary session of paper 1 of NET JRF in life science. So thank you for listening and all the best, everyone.